Hello students, myself Professor Dr. Raksh Dubey. I am a professor in the Department of Anatomy, Himalayan Institute of Medical Sciences, Swami Rama Himalayan University, Dehradun. As you have understood earlier as well that we are recording of various lectures on the human histology, and in that series again I am presenting today with the connective tissue histology. Well, in the earlier videos, if you have not seen them, first visit them and you will be understanding what about the sections of the histology and the epithelium, how they are there present. And now in the next topic, that is the connective tissue. Well, connective tissue, as the name itself is indicating, that it is connecting the various structures present into the histological domain in any organ, but frankly speaking, it's a filling material as well. It fills the gap in present between the various tissue islands and connect them with each other. It's a very vast structures, connective tissue. Basically, connective tissue gives modification to the bones, modification to the cartilages, and there are so many modification of this connective tissue. So let's start with the basic idea what connective tissue looks like and what are the basic constituents of this connective tissue or the filling material of the human histological structuring. As you see there are so many thread-like structure, blue structures are present, then there are the many pink color ropes are present here. Here we see the blue color dots. I have told you in earlier videos that this blue color is of the nucleus as well. And then various cells are present here. So it's a lot of messy structure is present over here, and let's see one by one what these structures are there. This pink color threads are named as collagen fibers. I hope you must be knowing what is collagen is. White collagen fibers you must have been taught in your classes. These white collagen fibers are very strong structures. They give the real strength to any structure, and there are certain cells are present over there, and these cells are the blast cells of this which forms the white collagens, and it is depicted into the blue color dot as well. Why blue color? Because the cell cytoplasm is clearly matches with to the staining of the collagen fibers, and you cannot differentiate if you do not using very high technique. It is almost impossible to distinguish between the cytoplasm of the fibroblast cells and the collagen fibers. So we see various blue color dots are present over here. Then we see this blue colored threads are present randomly, and these are elastic fibers. Connective tissue is a bunch of mixing of various structures. One organ or one fiber is giving you the strength, that is the white collagen fiber. On the another extreme, the elastin fibers are giving in the tensile strength as well as elasticity as well, and where it is visible into the histology like this type of blue colored threads. Then we see certain blood cells are present as here lymphocyte. Connective tissue are filled up of many sort of the white blood cell for the security purposes, for the purpose of the defense as well. So they are also present here. Then we see this special type of dot cell named as mast cell. Here we see in this picture the large lymphocytes are present, the macrophages are also present because they are eating up, and they are, you must be knowing, they are the couple of cells named as plasma cells, and various areas of inflammation and defense mechanism. These plasma cells are important. Here we also see one of the plasma cells. So various cells with fibers, if they are present, that is the connective tissue. Simultaneously there is another filling structure are present which are made up of complex protein and glucose structure, glycoprotein structures which are filling is these gaps where we see in this white color structure. So these all are making a filling jelly like structure, and that jelly is named as connective tissue. Now it is time to visit couple of the cells. What are the cells present inside and if they have any importance, and wow, let's see them one by one. Fibroblasts, if you see this the tongue shaped structures are present on this fibroblast. Basically these cells are producing white collagen fibers, and these fibers are coming out of these areas, and these are the legs or the hands sticking on that fiber. This is the fibroblast, a big nucleus centrally placed, almost oval in shape is present here, and the yellow color cytoplasm is there. Lots of mechanism is going on inside the cytoplasm, making the fibers, and these fibers are pouring outside, and that makes the various sort of fibers here. Second is the plasma cell. Plasma cells are very, very unique in their structure. If you see this, on the one side we see there is a blue color nucleus. Can you see this nucleus very closely? Can you observe these blue colored breakings in this nucleus? It is named as cartwheel nucleus, like cartwheel, just like spokes and the central area. Here we have this, and there are the various spokey structures are present here. It is named as cartwheel appearance of the mast cell nucleus. It's a very, very commonly and clearly visible structure. Whenever you observe a histology slide of connective tissue, if you observe the cell and if you see this nucleus in one go, you can understand it, it's plasma cell, and you will never forget in your life how does a plasma cell look like. Then you see this spatial structure that is named as adipose cell. On nucleus is almost stick to the periphery. The whole area is occupied with a vacuole, and that vacuole contains the fat. Whenever we take this histology slide, remember the fat is dissolved into the alcohol and taken out during the preparation. If you want to preserve that fat as well, then the technique is different and complex. But in H&E staining, we do not see this type of fat drops present inside the adipose. On that place, we see an empty vacuole structure occupying almost everything of the cell inside, and the nucleus is just shifted to the one side, and that gives a whole cell into a ring-shaped structure. Okay. Then we see the macrophages and the lymphocytes. They are the different lymphocytes. It's a large one, and lower side we see the small lymphocyte. The whole, as you know that lymphocyte, if you see it under the microscope, we see only a big nucleus. Very scary cytoplasm is present, just a cytoplasmic ring on the outside this nucleus, and we see on both sides small and large lymphocyte. Of course, these cells are used for the purpose of the immunity, where we see that there are the many macrophages are present which are eating off of this. These are the fibrocytes, and fibrocytes are the cell which are not forming the fibers and are present on the one side. Then blood cells, typically bilobe nucleus of the eosinophil we see here. Neutrophil multilobe nucleus is present. So if you have gone through your hematology classes into the physiology department, you must be knowing all these blood structures if you have analyzed the peripheral blood smear. Then there are the various pigment cells are also present in connective tissue, and into the cytoplasm. Different colors of the pigments are present. It gives you the various colors to the areas like the iris, like the skin. So other area, these are the cells which are present also into the cytoplasm. Mast cell, this containing various sort of chemicals that is used for the inflammation and other the defense reaction mechanism. So these are the cells which are chiefly present into the connective tissue. Fibers we have already discussed, white collagen and yellow elastin fibers. All these complex structures are present into the connective tissue. Remember, in one section it is not possible to visualize all this spectrum of the cells in one area. Also their composition changes at the area wise. In certain area, certain cells are more present, while in another area, certain cell numbers are reduced and another group of cells dominate here. So you have to analyze it from where you are taking the tissue and what are you are visualizing, or in a vice versa manner, if you are visualizing this one, what could be the organ from where this, or the area from where this connective tissue has been collected. So that is the basic idea of the connective tissue. I hope you must have understood how connective tissue is in histologically visualized. So we will see you in the next video with another interesting topic. Thank you.